Hello everyone and welcome back to another class, another video tutorial of the sexual reproduction in flowering plants. In this video, we are going to talk about the development of the embryo from the zygote. So the mechanism of development of the embryo from the zygote is considered as embryogeny or embryogenesis. How the development is taking place, before moving ahead into that, let me just draw the structure that once the fertilization event has occurred, what will happen inside the embryo sac. If this is the embryo sac, then with the zygote here inside the XII, the zygote have been formed, right? This is the zygote which have been formed here, and here the primary endospermic nucleus or the endosperm is present. Now endosperm is full of nourishment or the nutrients. So it is very important for the zygote. Zygote will show repeated mitotic cell division. So firstly the zygote will be divided into this kind of a structure. And then let me once again just draw the whole structure. This is the primary endospermic nucleus or the pen which is present there. So firstly the zygote will be divided into like this, and after that it will show repeated division like this, transversely, diagonally. They will start to repeat. They will show this kind of cell division. Okay, so by showing this type of cell division, after some time it will become somehow this type of a structure. There are also some cells which are present, and this type of a structure which will further show the cell division continuously. And here is the primary endospermic nucleus. Okay, so what is the reason behind this type of a structure? This is considered as globular phase of the embryo, and this is called as stalk or suspensor. This is called as stalk or the suspensor. And the head portion or the main part, that is considered as globular, as this is globular in shape. So why the formation of the stalk or suspensor is necessary, or why the cell will divide, or they will show this type of different types of cell division. They could have shown this type of longitudinal cell division also, but they are only showing the cell division like transversely. Why so? Because to give rise to the stalk or the suspensor. Why is it necessary? As you can see, that primary endospermic nucleus is farther away from this portion, which is the main part of the embryo. And as the primary endospermic nucleus or the endosperm is containing the nourishment, so what will happen? Until and unless it is reaching towards the endosperm, it cannot draw the nutrition from them. So to draw the nutrition from the endosperm, it is very important to make them reach to the endospermic cells. That is why the necessary of formation of stalk or suspensor is very, very important. Now, after showing repeated cell division, there will be the differentiation in the formation of the cells, like they will form this kind of heart-shaped structure. There are also the similar kind of cell division will be present. Okay, and here comes the endospermic cells. This is the heart-shaped stage, and right after reaching there, they will show further this type of growth. So these are the suspensor or the stalk. This is the structure which will now have reached to the endosperm to draw the nutrition in them. So in this way, the development of the embryo can happen from the zygote. Okay. So from the zygote, how the embryo can be formed, that I have already said and just now said, and that is the process of embryogenesis. Now let's talk about the endosperm development. There are also different types of endosperm development. So endosperm can be developed in 3 different ways. In one case, supposedly this is the cell inside which this is the primary endospermic nucleus. Say for example, this is the primary endospermic nucleus which is of 3N. That can be divided like this. By repeated division, they can form so many nucleus. So only free nuclear division will be seen here. What is the meaning of free nuclear division? What is the meaning of free nuclear division? That means right after the formation of the nucleus, there will be no formation of cell wall, cell envelope, nothing will be present there. That is why that is considered as free nuclear envelope or free nuclear endosperm, and this type of division is also considered as free nuclear division, and this type of endosperm is considered as nuclear endosperm. Okay, this is considered as nuclear endosperm. So where we can find free nuclear endosperm? In the tender coconut water. That is the foremost example of free nuclear endosperm. Next type is there where right after the formation and division of the nucleus, they can be surrounded with the cell membrane. They can be surrounded with the cell membrane or envelope is present there. They can be covered with the cell membrane. So that type of endosperm is considered as cellular type. And in the second type, in the third type, that is the mixture of the both of them, like nuclear and cellular, both of them are present there. In that case, that is considered as halobial. That means in few cases we can find only the formation of nucleus, and in the other few cases we can find the formation of the nucleus as well as the cell wall surrounding the nucleus. That means cellular and nuclear, both of them are present. So let me write it down here. Cellular and nuclear. Cellular and nuclear, both of the types are present in the halobial type of endosperm. So here I am going to wrap up my video today, where I have already talked about the embryogeny, that is development of the embryo from the zygote, and the development of the endosperm. Okay, so in my next video I am going to talk about the detailed structure of the monocot and the dicot seeds. So keep on watching my videos, and if you found this video helpful, then don't forget to like, share and subscribe to my channel. Thank you.